Hello, students, and welcome. Today, we're going to learn about one of the most important and, frankly, amazing molecules inside all of us. Let's begin today's explainer. But first, let us start with a question to create some curiosity. Just think about this for a moment. How is it possible that changing just one single tiny molecule in our body can cause a major, serious disease? It sounds almost impossible, no? Well, the answer to this question, and so many others about how our bodies work, is found in today's topic, proteins. You can think of them as the worker molecules of our cells. They are the ones doing all the hard work to keep us alive and healthy. Okay, so to understand all this, we have to start at the very basic level. Let us first understand what a protein is, its basic blueprint. Now, please do not get intimidated by these technical terms. At its simplest level, a protein is just a large polymer. Think of it like a long chain, like a necklace made of many beads. These individual beads are what we call amino acids. So, what is really happening here? These amino acids, these beads, they link together using something called peptide bonds. But what is truly crucial is the specific sequence, the exact order of these amino acids. You see, this sequence forces the entire chain to fold itself into a very precise three-dimensional shape. And it is this 3D shape that is the key to everything. So for your exam, this is the definition you should remember. A protein is a polymer of amino acids linked by peptide bonds. We call it a heteropolymer because hetero just means different. The chain is made of different types of amino acids. And the most important part, the specific sequence determines its 3D structure and its biological function. Now, here is a very common point of confusion for many students. It is very easy to think that proteins are just long, simple chains of amino acids. But the reality is, the chain is just the beginning. It's just the start. It is the complex, folded, 3D shape that actually does all the work. Please remember this. The shape is the function. All right, so how does this chain fold into a specific 3D shape? Let's look at the magic of protein folding. To understand this, we will use a simple analogy. I want you to imagine that each amino acid is like a small magnetic bead, okay? And each type of bead has its own unique shape and its own magnetic charge. Some are positive, some are negative. Now, what if you string these magnetic beads together in a specific order? Well, because of their unique magnetic properties, some beads will attract each other and some will repel each other. And because of all this pushing and pulling, the entire string will spontaneously, automatically fold itself into a very precise and complex 3D shape. It's just like a piece of origami paper folding itself. And this, students, is the main idea for today. This final folded 3D shape, this is the protein's function. It's not just some random shape. This specific structure is what allows the protein to do its job perfectly. So why does this concept matter so much? Well, because this single idea that structure determines function, it explains so many things. It tells us how genetic diseases like sickle cell anemia happen. It helps us understand how our antibodies fight infections. It even explains why heating food changes it. This idea is the basis for so much of modern medicine. Okay, enough of theory. Let us look at some practical, real-world examples that you can actually see. This will make the concept absolutely crystal clear for you. So an observable phenomenon you see almost every day is cooking an egg. You start with a clear egg white, right? That is mostly a protein called albumin, all folded up in its natural state. When you apply heat, you break the weak bonds that are holding that 3D shape together. The protein unfolds, and all the unfolded chains get tangled up with each other. This is what turns the egg white solid and opaque. This process is called denaturation. And this brings us back to the question we started with. In a disease like sickle cell anemia, what happens is that a single amino acid, just one, glutamic acid, is changed to another one, valine. This one tiny change alters the final shape of the hemoglobin protein. And because the protein's shape is distorted, the entire red blood cell becomes distorted. It's a perfect example of how one small change in the sequence has such a big effect on the function. So, to summarize our discussion, what are the most important takeaways? What are the key points you must remember? You just need to remember these 3 things. First, there is the blueprint, which is the sequence of amino acids. Second, this sequence causes the protein to automatically fold into a specific 3D shape. And third, this unique 3D shape is what gives the protein its function. So the flow is simple. Sequence leads to shape, and shape leads to function. So if you just remember the blueprint, the fold, and one real example like the cooking of an egg, I can assure you will master this topic. It will feel much, much lighter in your exam. Keep studying hard.